My name is Dan Roxer. I'm a professor here in molecular and cell biology, and I'll be your master of ceremonies today. I'm also the co-director of our NIH training grant in genomics and computational biology, which is the organization that sponsors this event. And for the last 4 years, we've had gatherings in January to look at different aspects of genomics with an emphasis on human biology. In the past, we've worked on human genetic variation, genomic medicine, cancer genomics, and this year, genomics in the brain. Just by way of introduction, there's a noted British astronomer, Martin Rees, and he's said many times that there are three big frontiers. There's the very small, the very large, and the very complex. And so physicists and astronomers kind of have a monopoly on the very small and the very large, but biologists have the very complex in the brain. And what we're going to learn about in the next day is different aspects of approaching brain function, development, and disruption using the tools and approaches of genomics. So like many complex systems, there's a certain amount of robustness, and we know that with a lot of genetic variation, we see variation in personality and behavior, but we all kind of recognize the normal range. But of course, like any complex system, if you disrupt it too much, you can break it. And so there's a big also a big focus on neuropsychiatric disease and the ways in which genetics and the environment can have an interplay on loss of function. In addition, there's also a growing appreciation that the brain is very plastic, both in its synaptic connections, but also in its genome itself. And we're going to hear today about both epigenetic and transposable element mediated variation in the genomes of different brain cells. So I think it's going to be very exciting. Our first speaker is Megan Dennis. Megan, I was hoping to be the first person to introduce her as a new faculty member at Davis, but that's not starting until this summer, so I make believe anyway. So Megan is finishing her postdoctoral studies with Evan Eichler at University of Washington, where she's been a leader in understanding the role of segmental duplication and copy number variation in brain function with Evan. Before that, she earned her bachelor's degree in biochemical engineering from Michigan State, and her PhD jointly at the University of Oxford and at the NIH, working with Tony Monaco and Eric Green, looking at various candidate genes for autism, I think. Is that right? Dyslexia, excuse me. They're appropriate. She's also the first author of a paper that has really been important for me. When I found it, pretty much it was the only thing we talked about for weeks in my lab, where she looked at the role of a specific gene duplication in changes in brain development from the ancestral apes to humans. And that's been a real interesting way of thinking about the way small evolutionary changes can have a big impact. So Megan is going to give a talk today, and you have your the role of segmental duplications in human brain evolution and disease. Thanks, Megan. I'm sorry.
Great. Thank you so much for the invitation to speak here. It's truly a pleasure to talk to you today about some of the work that's ongoing as a postdoc in Evan Eichler's lab and looking at the role of segmental duplications in human brain evolution. As Dan mentioned, I'll be starting as a faculty at UC Davis in the summer, so I'm very excited to get that started, kind of continuing on work that I've done as a postdoc, and probably exploring some new questions as well. So let's get started. So my overarching interest through grad school and up through now has been to try to identify genes that contribute to human-specific neurological traits and disease. And so if you just take a look at a human and a chimpanzee, we can see that there's lots of phenotypic differences, obvious ones such as body hair and the way that we walk and the way that we interact. But underlying, there are also neurological differences. And so there are differences in the size of the neocortex. So within the last 6 to 8 million years, the humans have had an expansion of the neocortex. Also, there are distinctions in synaptogenesis, such as the timing at which the maturation of synapses occur, as well as the actual physical appearance of the synapses, such as the increased density of dendritic spines. So there's been a lot of research that's gone into trying to identify the underlying genetic components contributing to these neurological traits, just looking at, typically looking at single nucleotide variants. We can specifically look at genes that are undergoing human-specific positive selection by looking at differences in the protein coding variants. And so one example of this has been FOXP2, in which we see evidence of human-specific positive selection on a language gene. And so single nucleotide variants account for about 1% of differences between human and chimpanzees, but it can't account for all of the phenotypic variation that we find between human and chimpanzee. At least we haven't been able to find the causes of these phenotypic differences. And so we propose that another source of genetic variation that accounts for an even larger amount of differences between humans and chimps and also across humans generally are segmental duplications. And this is a focus of the Eichler lab. And so it accounts for over 2.5% of differences between human and chimps and over 5% of variation across human populations compared to 1% in humans of SNPs. And so we define segmental duplications, and I'll be showing them for the rest of my talk as these colored arrows. And so we define them as large genomic segments that are greater than 1 kb in size that share a sequence identity of over 90%. And so shown here is a chimpanzee segment that's not duplicated, and then in humans it's been duplicated. These type of regions are not amenable to standard genetic analyses because they're so similar to each other, and they're often missing or incorrect in the human reference genomes. Further, we often ignore these duplicated segments of the genome because our algorithms typically can't really look at them. And so for instance, we can take a mapper such as BWA, which takes these short read sequences and maps them against a reference genome. And one of the filters that is commonly used is to throw away any reads that map to multiple portions of the genome. And these are precisely the reads that we would be mapping to these segmental duplications. And so when we look at these genome-wide studies, we are often overlooking the genes that are within the segmental duplications. And so during my time as a postdoc, I focused specifically on a particular class of segmental duplications, and these are the ones that have arisen in the last 6 to 8 million years, so after the human-chimpanzee divergence. And so because of this, they're even more difficult to kind of assess because they're over 98% similar to each other. They haven't had very much time to accrue mutations that allow us to differentiate between these duplications. And so if we actually take a look at the human reference genome, a lot of the gaps that are still existing in the GRCH38 build are in these segmental duplication regions. And so what's interesting about these regions is that they are enriched for expressed genes. And so for us, that's quite exciting because it makes them a fertile ground for the birth of new genes with novel functions. And so just giving one example here, we can have our ancestral locus, which has gene 1 and gene 2, and when we create this duplicated human-specific version, it allows for the creation of just a complete gene to be duplicated and have the same function or undergo neofunctionalization, subfunctionalization. But we can also have cases in which at birth we can have a new function, such as creating a truncated gene that may have different cis-acting elements that would cause the gene to be expressed in novel tissues. And we can also plop a gene right in the middle of another gene and create gene fusion events. And so this is really a mechanism that could create novel function immediately. And so recently, by comparing recent whole genome sequencing from the 1,000 Genomes Project with sequences from other great apes, we've been able to identify 25 gene families that are uniquely duplicated in humans. So that's at copy number 2, diploid copy number 2 in other great apes, and have been specifically expanded in humans. And it accounts for about approximately 60 gene paralogs. And so I've just shown some of them here on these human chromosome ideograms by these, they're indicated by these blue arrows. And so excitingly, these human-specific duplicated genes are enriched for genes with neurological function. And so I'm just showing you some of examples here. So we've got this SRGAP2, which is involved in neuronal migration on human chromosome 1, Haydn, in which knockouts in mouse contribute to hydrocephalus, and there have been studies in which it's been maybe implicated in autism. SMN1, which is a gene, when mutated, causes spinal muscular atrophy. GTF2-IRD2, which is a transcription factor important in brain development. We have GPRIN2, a GPRIN inducer of neuron outgrowth, as well as TRNA7, which is a gene in which deletions of this particular gene are thought to contribute to epilepsy. And so I'm going to talk briefly about the SRGAP2 story for some of you who may not be familiar with it, or just to come back and summarize it for you guys. So this gene was, we were interested in characterizing this gene initially from our list of 25 genes because it had already been characterized by another group, Frank Pelot's group, who's now at Columbia University. And so it was functionally characterized in mouse, and it was found to be contributing to neuronal migration and morphogenesis by working in concert with other proteins such as F-actin at the cell membrane surface and neuronal precursor cells. And so there are 3 functional domains. There's this FR domain, which is responsible for the homodimerization of the protein at the cell surface. And then these other domains, rho-GTPase and SH3, which work in concert with others at the cell membrane surface. And so I want to just draw your attention to this FR domain, which will become important later. And so the reason we thought that this was a good gene to kind of start out with was because they found that levels of SIRGAP2 contributed to altering levels of neuronal migration in these mice. And so what I'm showing you here is just a cartoon of neuronal precursors undergoing neuronal migration. So that these neuronal precursors are in green, and they typically start, well, they start in the ventricular zone, they attach to glial fibers, and they travel upwards towards the cortical plate. And so what Pelot had suggested from his studies was that SIRGAP2 was capable of inducing philopodia within these neuronal precursors, and in fact, turning on the brakes for these neuronal precursors. And so when they have overexpression of SIRGAP2, we have philopodia that are induced too soon, and then we have neuronal precursors that don't go far enough in the cortex, and then if you knock down SIRGAP2, we have these neurons that migrate too far and too fast. And so for us, we thought this was a very interesting gene to begin with. And so to begin, we wanted to understand the nature and the duplications in humans. And so what I'm showing you here are fluorescent in situ hybridizations of human, chimpanzee, and orangutan. And so what we found was that SIRGAP2 was indeed duplicated specifically in humans compared to the chimp and orang. We've got the SIRGAP2A, which is the ancestral version, on human chromosome 1q32, and you can see it's shared between chimp and orang as well. We've got these 2 human-specific versions, SIRGAP2B and SIRGAP2C, that flank the centromere of human chromosome 1 at 1q21 and 1p12, and they are not seen in these other great apes. And when we take a look at other mammals, we find that SIRGAP2 isn't duplicated in other mammals as well, so it's not like it's some gene that's just commonly duplicated that we see in vertebrates. It's actually unique to humans. And when we took a look in the human reference at the time, which was HG19, GRCH37, we didn't find all of these versions in the reference. And this is something that we commonly see because these duplicated genes are so similar to each other. In fact, even the ancestral SIRGAP2A had a gap right in the middle of the gene. And so why is it that we have this problem with sequencing these genes and assembling them in the human reference? And so the primary problem is that the reference was built from a diploid genome, or multiple diploid genomes. And so with these paralogs, we are struggling with trying to discern between allelic sequence variants, so SNPs, versus prologous sequence variants that exist that distinguish SIRGAP A, B, and C. And so to get around this problem, our lab and others have developed a haploid back resource in which we no longer have to work with a diploid genome, but instead we only have a haploid genome, and so we only have to work with paralogous sequence variants. This back library was created from a hydatidiform mole, which is the product of the fertilization of an enucleated egg. And so this particular cell type only has the genome coming from the father's sperm. And so this mole back library is called Cori17. It's actually publicly available, and we have back-end sequences that are available for it as well that have been mapped back to the reference genome. And so we used this library with the back mappings, and we just looked for concordant mappings across our genes that were available in the reference genome, and we also looked for discordant mappings. So we had mappings where only one end was mapping and another end was not mapping on the reference, and so we just took all of these clones and we performed capillary sequencing in collaboration with WashU. And by leveraging these paralogous sequence variants that distinguished our various paralogs, we were able to create these supercontigs that span SIRGAP2A, B, and C. And so overall, we were able to identify 380 kilobase of sequence that was completely missing from the reference genome, and over 500 kilobase of sequence that was corrected. So either we had a particular back that was in the complete wrong portion of the genome, or it was in the incorrect orientation. And so when we actually take a look at these contigs and try to understand the nature of the duplication, I'm showing you here a mirror-repeats image in which we have homologous segments connected by these colored lines. We see that SIRGAP2A and SIRGAP2B share 244 kilobase of sequence. And so that shows that there was a duplication of 244 kilobase that came from A and established B. And within this duplication, we have this FAM72A gene, which is an uncharacterized gene that's small, as well as the first 9 exons of SIRGAP2A, as well as any kind of putative cis-acting regulatory elements. And so what this suggests is that if we actually have a transcript that's being expressed from SIRGAP2B, it would be predicted to encode a truncated SIRGAP2 copy that would encode for nearly the entire FR domain, so this domain that's responsible for the dimerization of the protein. When we take a look at the similarities between SIRGAP2B and SIRGAP2C, we see that there's an even larger shared sequence, so over 500 kilobase of sequence, and that suggests that an even larger secondary duplication occurred that established SIRGAP2C across the centromere. And so this SIRGAP2C also contains FAM72 and all of the first 9 exons of SIRGAP2A. And please, I'm actually quite happy to take any questions if at any point something isn't clear, and so just feel free to interrupt me if you'd like. And so because we have these homologous sequences, we were able to perform timing estimates by creating a phylogenetic tree from the alignments and pulling out paralogous sequence in chimpanzee and orangutan. And from these timing estimates, we predict that the initial SIRGAP2A to SIRGAP2B truncated segmental duplication occurred approximately 3.4 million years ago, assuming that there's a divergence of chimp and human of 5 to 7 million years ago. And then we had the secondary duplication that occurred about 1 million years later, which established SIRGAP2C. And so to summarize up to this point, we had this full-length SIRGAP2A that was duplicated about 3.4 million years ago and created this truncated SIRGAP2B at 1q21. And this was an incomplete segmental duplication that would have created this truncated gene at birth. And then it was followed up by the secondary duplication about 1 million years later to create SIRGAP2C. And for us, just thinking about looking at fossil records and looking back on the evolution of humans, this placed the duplication in a time in which our ancestors were undergoing a lot of phenotypic changes, including the expansion of the neocortex. This duplication here was following the divergence of Homo sapien from Australopithecus, and so this was a very exciting time for our ancestors, although I'm not saying that SIRGAP2A is responsible for all of those changes, but it places it in a time that would be quite exciting. We also took a look at the Denisova and the Neanderthal genomes, and we found that both of these genomes contained all of these SIRGAP2 copies, and so that just backs up our timing estimates for our SIRGAP2 copies. And so we wanted to get a sense for today, do all humans have these particular duplicated copies? Because if you have a gene if you're looking for genes that contribute to human-specific traits, you would expect every human to have that particular gene. And so to get at this, we leveraged the paralogous sequence variants that existed between SIRGAP2A, B, and C. And we took 1,000 genomes data, so we genotyped 661 individuals, and did read depth estimates across these paralogous sequence variants. And so what I'm showing you here are SIRGAP2A copy numbers against SIRGAP2B. And so we see here that SIRGAP2A is fixed at copy number 2 across all 600 individuals. And this is something that we were expecting because this is an ancestral conserved gene that we see in all mammals, and we know it has a brain function. But we do see that SIRGAP2B is copy number polymorphic, ranging in copy number from 0 to upwards of 4. And so that suggests that this gene isn't indeed the gene that makes us human in any way, because there are normal individuals walking around without any copies of SIRGAP2B. But surprisingly, when we look at SIRGAP2C, the granddaughter copy, so the more recent duplicated copy, we do see that it's fixed at copy number 2 across all humans that we've tested. And this is quite remarkable considering that if we look at the landscape in which SIRGAP2C falls, it falls within a region that's highly duplicated. And so what we often find in these kind of regions that have lots of segmental duplication is that they're pretty tumultuous. And once you have one duplication, you can kind of easily delete or duplicate things. And so the fact that it's really fixed at copy number 2 to us was very interesting and suggested to us that this could be a candidate for function. And so to get at this question, we first wanted to know, is SIRGAP2C or B or A, are they expressed, and in what tissues are they expressed? And so I just did a simple reverse transcriptase PCR shown here, and we can see that the ancestral version is expressed in human fetal brain, and the B version is expressed as well as the C version is expressed. From this analysis, we were able to sequence these transcripts and find that there was a 5' or a 3' UTR that was unique to the duplicated versions. and leveraged this information in collaboration with Frank Pelot's group, who had published a paper at the exact same time we did, who focused on the functional characterization of SIRGAP2C. And so what they found from doing in situ hybridizations in human embryonic brain at gestational week 12 was that SIRGAP2A was expressed highly in the ventricular zone as well as upwards in the cortical plate, and we see a very similar expression pattern for SIRGAP2B and C. And so this particular probe isn't able to distinguish between the B and C copies. And so this is something that we were kind of expecting because we did know that the cis regulatory elements were duplicated. And so this just added to the fact that we believe that these genes are actually co-expressed with each other. And so the Pelot group went ahead and did a ton of other functional experiments, and so I'm just going to talk a little bit about what they found, looking at the function of SIRGAP2A and SIRGAP2C. And so this is just one assay that they do in which they show that SIRGAP2A induces philopodia when it is dimerized at the cell membrane surface. And so when they overexpress SIRGAP2C, you can see clearly that these philopodia are induced. And so when they co-express SIRGAP2A with the duplicated SIRGAP2C form in these mice, or in this cell line, they find that these philopodia are markedly reduced. And so what they were able to deduce from this was that SIRGAP2A and SIRGAP2C were acting antagonistically, and the mechanism by which they suggest it happens is via heterodimerization of this FR domain. And so what we have is we've got this full-length functional SIRGAP2A that's able to assemble at the cell membrane surface. SIRGAP2C cannot because it doesn't have these other functional domains, but it can dimerize to SIRGAP2A. And so essentially it's usurping functional SIRGAP2A from the cell membrane surface and causing less philopodia to actually be generated. And so you can imagine that these decreased philopodia could allow neurons to migrate faster during cortex development or farther. And so this could have contributed during the expansion of the neocortex. Further, they did a ton of other studies in which they show that SIRGAP2C overexpression in the mouse and also in cell lines leads to human-like synaptic features in these mice. And so they find that the timing of synaptogenesis is much more delayed, and this is a feature that we have in humans, as well as an increased density of dendritic spines on these synapses. And so overall, these combined results suggest that the human-specific segmental duplication SIRGAP2C, this granddaughter copy, may have been important in human brain evolution, and perhaps in part by contributing to the expansion of the neocortex, as well as contributing to unique synaptic features in humans. And so I've shown you an example of SIRGAP2 in which segmental duplications can lead to the creation of novel genes that can contribute to novel neurological traits. But on the flip side, we also see that these same segmental duplications that contain these novel and exciting genes also sensitize humans to disease. And that's by sensitizing our genome to large-scale deletions and duplications. And so this occurs specifically in these regions we call genomic hotspots. And so we have these segmental duplications, if they're in direct orientation with each other, can undergo non-allelic homologous recombination during meiosis and can lead to large-scale duplications and large-scale deletions. And approximately 15% of developmental delay is caused by these large-scale CNVs, and the majority of them are occurring recurrently within these genomic hotspots. And so there's this strange dichotomy in which these segmental duplications in humans could be helping us in creating innovation, but on the same side, they've also sensitized us to disease. And so there's approximately 120 genomic hotspots in the human genome, and I'm showing them here by orange bars. These are all hypothetical genomic hotspots, because basically we just did an assay of the entire human genome and looked for regions in which segmental duplications were directly oriented. And if we actually overlay some regions that we know are associated with disease, there are already a number of diseases that have been implicated with these genomic hotspots, including the 1q21 deletion and duplications that are associated with autism and intellectual disability. We have the Williams-Barian syndrome region on chromosome 7, 15q13 microdeletions, which I'm going to talk a little bit more about later. The Prader-Willi-Angelman syndrome, as well as the 16p11.2 deletions and duplications, which account for approximately 1% of autism cases. And so to systematically understand the role that these genomic hotspots play in a specific neurological disorder, so in autism, we designed and ran a custom-designed Agilent array that targeted specifically these 120 genomic hotspots to understand if any of them were associated with disease on a systematic level. And so we did this for 2,500 autism probands, consisting of over 2,000 probands from the Simon Simplex collection, which is composed of kids who have simplex families, so kids who have autism and parents who don't have autism, as well as nearly 600 degree probands, which come from multiplex families. And so when we look at these 120 genomic hotspots, we find that there are about 25% of the hotspots which actually do have CNVs, so the other 75% we couldn't find any CNVs, even in cases or controls. But overall, there is an enrichment of CNVs in our autism cases, about 3% compared to 1.3% in controls. And we compared our CNVs to a control of about a cohort of 8,000 population controls. We take a closer look at the specific genomic hotspots that are coming up that are associated with autism compared to controls here. So autism prevalence versus controls in green. We see that there are these regions that have been implicated previously, including the 16p11.2 duplication and deletion, the 15q13 deletion, and also the Williams-Urine syndrome duplication. But we did find one novel duplication, which was 1q21, which hadn't been previously associated with autism at the time. And we also take a look at specific genic events in the genome. And we find that there are human-specific duplicated genes that are also being affected by these CNVs that are potentially contributing to disease. And so this is just one example in which we find large-scale deletions and duplications affecting hydin, which was a gene that's been found to be important, or in knockout mice contributes to hydrocephalus. And so we see this large-scale deletion. So this is a microarray plot, and the red indicates a deletion, and the blue indicates a duplication, and hydin's located here. So this is a very large-scale de novo deletion in one SSC, simplex proband, and then we have this large-scale duplication here and here. And so this was quite exciting for us. It suggests that these human-specific duplications could also be contributing to neurological disorders. And in fact, when we take a closer look at the locations of the genomic hotspots, we overlay now the genomic hotspots in orange with the human-specific duplicated genes, we find that a lot of these genes are falling within these regions that are associated with disease. And so we wonder if these genes are contributing to the phenotypes that we see here. And certainly these genes are contributing, the segmental duplications that they exist in are contributing to the actual CNV events that are occurring. And so it's something we'd like to systematically get at, and so for the last couple of years, we've been systematically going through and sequencing all of these genomic hotspot regions that contain these human-specific duplications using the CH17 back resource to try to get a better sense of the mechanisms by which these are occurring, and actually the gene content underneath. And so instead of doing the capillary sequencing, we're now using PacBio sequencing, which is way faster and cheaper. So we can sequence a bat clone within a couple weeks for about $700. It adds up for sure, but it's certainly faster and cheaper than capillary sequencing. And we can get very nice, high-quality de novo assemblies from that. And so I'm just going to talk a little bit about one particular region in which we've performed a ton of sequencing and show you one example of what we can do with this back sequencing resource. And so we are interested in looking at the 15q13 microdeletion region. If we take a look at the human chromosome 15, we said that there's a ton of genomic hotspots that are associated with disease, but specifically this human 15 Q13.3 shown here is one of among the most common microdeletion syndromes, and so it accounts for 0.3% of cases with developmental delay, as well as 0.2% of cases with epilepsy, schizophrenia, and autism. And so I am just showing you the underlying segmental duplication landscape of this particular locus. And so we've got breakpoint 4 and breakpoint 5. There's a 2 megabase deletion that spans between this cluster and this cluster of segmental duplications. And we've got these 3 segmental duplications shown here. So there's this, what I'll call alpha, which is in green, beta, which is in red, and this orange one, which I won't really talk about. And just by looking at this initially, we see that alpha and beta are in inverse orientation, so it's not exactly clear how NAHR is occurring, which then can lead to deletions and duplications of this locus, just by looking at the human reference genome here. And so we can go ahead and go back to our 1,000 genomes resource and do copy number estimates to try to understand the variation that we see at this locus across humans. And so alpha is about 300 kb in size, and it ranges in copy number from copy number 2 to copy number 5. It's a human-specific expansion, so all chimps have a diploid copy number of 2. So that means if you were looking on here, chimps would only have 1 copy. And then the majority of humans have 4 copies of alpha. So that's what we see in the human reference. This is considered 4 copies because it's 2 copies on 1 chromosome. We see that beta also is a human-specific expansion, ranging from 5 to 8, but there are other versions of beta elsewhere in the genome. And so the majority of individuals have copy number 6 to 7, and that's what we see in the human reference. And so by combining these copy number estimates with fluorescent in situ hybridizations that we performed on various individuals with different copy numbers, we were able to discern that there were a multitude of haplotypes that existed in human populations today across this region. And so this is the human reference. Then this is a chimpanzee, and so we see that alpha and beta both exist at breakpoint 5, and there's no segmental duplications at breakpoint 4. And then we have these 3 human haplotypes, and so this is the simplest one, in which we have a beta inversion in the ancestors after the divergence of human and chimpanzee. And then we have this alpha-2, which is the reference. And then we have this 3 megabase haplotype, which also has an expansion of alpha and beta at breakpoint 5. And so just by looking at just normal 1,000 genomes populations, we see that there's an incredible amount of diversity just in this region alone. I mean, we're ranging in size from 2 to 3 megabase, so 1 megabase of sequence in a normal individual. That's different. As we often see in these rich segmental duplications regions, we also see inversions, and so we found this beta inversion at breakpoint 4, and we also identified this gamma inversion, which is large-scale 1.7 megabase pairs, spanning from breakpoint 4 and breakpoint 5, and that had been previously found. We just were validating that same one. And so this beta inversion we identified using the CH17 back library resource, and without using backs, it'd be very difficult to identify using kind of any other sequencing methods. And in fact, it's very difficult to identify this using fluorescent in situ hybridizations as well. And so to get at kind of the way that we do this, I'm showing you the various backs that we sequenced across the CH17 library for the 15q13 locus, so they're just from top to bottom. And then these right here are the Illumina read pileups against HG19 of the back reads. And so we can see that we have normal backs across here, but across the beta, we have these discordant backs. And so if you were to flip beta, we can see that there is indeed an inversion of this beta segmental duplication. And so we went ahead and performed both capillary and PacBio sequencing of all these clones, and to get a sense for this alternative haplotype and actually what was underlying this beta inversion. And so if we zoom in on that beta right there, and we compare, this is again a me-repeats image, we see that the beta segdup actually is composed of smaller segmental duplications within it. And so the beta segmental duplication inversion is mediated by these 60 kb inverted segmental duplications shown here by these light blue, and which contain this GOLGA 8 gene. And so the inversion breakpoints exist within these segdupes here, within this, what we call this GOLGA 8 core duplicon. If we take a look at the copy number of this particular duplicon, we find that it's over 30 in humans. And so this is actually a duplicon that is kind of it's just kind of spread all over chromosome 15. And it had been previously implicated as a primate-specific expansion that's associated with numerous intrachromosomal events, not only in humans, but also in other great apes, including likely mediating large-scale inversion in chimpanzee. And if we actually map all the GOLGA duplicons at the 15q13 locus, which I can show them all here, we see that they are on the edges of all these segmental duplications. And so we wanted to get at the actual underlying sequence and see if truly these GOLGA duplicons were mediating these events. And so to do that, we sequenced over 100 clones across all these various human haplotypes. So we generated back libraries for various different humans, and we also took publicly available backs from orang, gorilla, and chimpanzee and performed sequencing, either Illumina sequencing or PacBio sequencing. And from this, we were able to precisely define the mechanisms by which these segmental duplications were arising. And so we see here that the beta inversion between chimpanzee and human was mediated by these GOLGA duplicons, as well as this alpha-beta duplication from breakpoint 5 to breakpoint 4. This beta inversion that I talked about already. We also have this expansion of alpha and beta at breakpoint 5, mediated by GOLGA. And then finally, the gamma inversion even is mediated by the GOLGA. And so we wondered if these same GOLGA duplicons could be contributing to the microdeletions that we see in patients. And so to get to this question, we identified 2 probands from the Simon simplex collection who had large-scale deletions at this region. And so I'm showing you an array CGH plot. And so basically we see that the deletion spans from breakpoint 4 to breakpoint 5. But because of the nature of array CGH, we can't actually discern the precise breakpoints within the flanking segmental duplications. And so we performed whole genome shotgun sequencing using Illumina of these 2 probands as well as their parents and other control samples, and looked at read depth across the region of the reads mapped back to the reference, and also to our CH17 contig. And we're able to delineate precisely the breakpoints to occur, again, within these GOLGA duplicons. And so, as I mentioned before, we had these inverted beta segmental duplications, but actually within the beta, we have these directly oriented smaller duplicons. And so if we take a step back and look at all the duplicons that exist on human chromosome 15, we see that they're spread all along, and in fact they are on the edges of all of these microdeletion hotspots that we see. And so it's very likely that these same GOLGA duplicons are contributing to the deletions and duplications that we see at these other genomic regions that are associated with disease. And there are different high copy duplicons that exist on other chromosomes. So for chromosome 15, we have the GOLGA duplicon, but for other chromosomes, we have different genes and different high copy duplicons that have expanded specifically on those chromosomes. And they could very likely be contributing to those as well, to those events as well. And so in all, I've talked to you today about how segmental duplications are likely important in evolution by not only creating these novel genes that could be contributing to remarkable neurological features in humans. And so I talked about SIRGAP2. And they are also important in that they cause disease. So they have these large-scale deletions and duplications that can lead to autism and intellectual disability, as well as epilepsy and other disorders. And mechanistically, these high copy number segmental duplication, or these duplicons, are likely important in establishing both these disease states and also these evolutionary events. And so we have a number of ongoing projects in the Eichler lab, and also I have some future directions, and I just probably will go through those with you now. And so for the first time, we have this gold standard genome, these gold standard genomes that now include all of the human-specific segmental duplications and their genes. And so we can now go back and systematically understand the role that these play in evolution and in disease. And so to start, we can start taking a look at the evolutionary history of these genes, and so when exactly they've arisen, and if there's any kind of interesting we can look at the evolution by which they occurred, and if there's anything interesting there in terms of timing. We can look at signatures of selection of these genes. So once they've arisen, are they undergoing neutral processes, or are they under purifying selection or positive selection? And also we can look across human populations today to get at that question. And so we have a number of exciting genes, including this BOLA2, which is the only gene of all these genes that has arisen after the Denisova Neanderthal human split. And so that one could be very interesting to look at that one. We can also try to understand if these genes are expressed, and if they're expressed, do they are predicted to encode gene models? Are they creating protein? And so this is something that's happening systematically, and in what tissues are they being expressed? And so this is something we were able to do for SIRGAP2A and B and C using the paralog sequence variants, and we are continuing to do that with these other genes. We'd also like to understand if mutations in these duplicated genes could lead to a disease state. And so this is something that has required us to try to come up with new methods, because we have these duplicated genes. They're not targeted by exome sequencing, so we often, actually, if you look at the exome capture, we often are missing these exons from the capture, and then they're not being mapped and they're not being characterized. And so we're going back and doing targeted sequence capture, paralog sequence variants, as well as the exons, to try to understand if these mutations contribute to any kind of neurological disorders. And so this is just an example of one of the methods, and so we can do targeted capture of the paralog sequence variants across SIRGAP2, and we can find large-scale deletions of SIRGAP2A, and then small-scale deletions of SIRGAP2C. And so this is a project that's ongoing, and in fact we are finding that there are in fact small-scale deletions of SIRGAP2C even in the normal population, and this is something that we weren't able to find before. And so this might suggest that SIRGAP2C may not be as important as we initially thought, but what we're finding is that in disease populations, we're finding homozygous mutations, and these homozygous mutations are not being found in the control populations. And so we're just going back and doing deep sequencing of tons of disease cohorts and as many controls as we can get at. And then finally, something that I'm personally interested in doing is trying to do functional characterization of some of these genes. And so we'll be doing molecular and cellular assays, kind of in the same line as these Philopodia assays that the Flow group did for SIRGAP2, and setting up a zebrafish resource. And so we can think about it's pretty funny to think about using zebrafish to look at human-specific neurological traits, but I think it's a good starting point to understand what the functional role of these ancestral genes are, and then potentially trying to humanize zebrafish by overexpressing the human-specific forms, and then from there potentially taking it to a different model system. I'd like to wrap up by thanking many people. Xander Nuttall, Santosh Girirajan, and Francesca Antonacci I worked in a team for the project that I presented today. Also, I'd like to thank Evan Eichler, my mentor, as well as all of you for your attention. I'm happy to take any questions now.
Thank you, Megan. Questions? Tom, why don't you lead us off?
You mentioned SIRGAP2C, I think it was, that it antagonized SIRGAP2A, but what about SIRGAP2B? Because it was actually quite similar. Would that overexpression also antagonize?
Yes, so SIRGAP2B, there were functional experiments done on both B and C, and they seem to do the same thing. So it's certainly possible that SIRGAP2B has a function and it could be contributing to variation that we see across human populations. One thing that we looked at was the overall level of expression of SIRGAP2B and C, and we found that C was much more highly expressed compared to C was more expressed over B. One of the reasons we thought that this might be the case is that if you were to look at the homologous sequences of B and C, we saw a number of really large-scale deletions upscale of B that occurred after the duplication to C. And we wonder if there's cis-acting elements there that just got rid of those particular regulatory elements that would have been important.
Difference in SNP frequency between the SIRGAP2C and B? 'Cause B had such a range of variation in humans compared to C. Was there any difference in the actual sequence polymorphism?
Yeah, so we're starting to get at that question. So it's pretty difficult to look at short read sequences and assign them to a particular paralog. When you have Illumina sequencing, you have to make sure that you have a paralogous sequence variant and also a SNP on that same read. And so this is something that, using the capture method, we're starting to understand the underlying SNP variation that exists between different paralogs. But it's as we also gain longer reads and we also hopefully come up with targeted methods to use PacBio sequencing, I think this is a question that I personally would be very interested in looking at, is looking at the variation. So also looking for signatures of selection that would maybe differ between the paralogs. So yes, yes. Yeah. Absolutely.
Tom pointed out that this dramatic variation in the SRGAP2B
Right.
could be very meaningful. They may all have two eyes and
Right.
four appendages, but in terms of intelligence or behavior,
Exactly.
prone to violence or whatever. So who is doing the detailed
Right. And so
psychological and neurobiological phenotyping of these so that we can make sense of what they do?
Can you rephrase the question?
Sure, yes. So what you're saying is that, we can find these variants that are occurring within these paralogs, but like what is the phenotype that that's connecting to? And so, while we're doing, we have this collaboration with a number of clinicians, and so typically what we do is from the beginning, just genotype first, we look for events, and if we find events, we go back and through our collaborations with clinicians are able to recontact families and get a deep phenotypic workup. And what we're finding actually with the SIRGAP2C cases is that there seems to be similar phenotypic features. And so what we see in SIRGAP2A is because SIRGAP2A has been associated, or there have been CNVs found in patients, and what we often see are abnormalities in the corpus callosum, and so this is one of the phenotypes that we're looking for from imaging studies, for instance. We also see that in, there are patients that have seizures, and so we are starting to see that this might be a unique and kind of common thread between them, but certainly I completely agree with you. We don't know what phenotypes we're looking for. We're coming from the gene first, and so that's very important that we actually get a deep phenotypic workup of all our patients. Yes. Sure, yes.
Could you say a little bit more about how you date the segmental duplications of the SIRGAP2 genes from the phylogenetic analysis?
Sure, how we did the dating of them? Sure, yes. So we did molecular timing estimates, and so basically we did we did multiple sequence alignments of SIRGAP2A, B, and C in human, and also pulled out the orthologous regions in chimpanzee and orangutan. And so by comparing the differences that we see along the chimp lineage between the common ancestor of chimp and human, and also looking at the differences that are accrued between the various paralogs, we can do molecular timing estimates, assuming from the fossil record that human and chimpanzee diverged about 5 to 7 million years ago. And so what you do is you basically do a comparison of the year. You have to get a molecular timing estimates using the genetic differences between the orthologs and the paralogs. So.
Adam,
So I have a question about.
could you give him a mic?
Question really, it's for when you look across the human genome, especially the GOLGA example, it looks like pretty clear that those regions of duplication are specific to humans. And I'm curious if in mouse there's enough data to figure out what regions of the mouse genome may be similar enough to ask what genes are under selection for mouse and other species and see whether or not you could take a method you apply to humans and apply it to others.
Sure. So let me rephrase that. So you'd want to know if we could use similar methods that we're using for humans to other organisms, including mouse. So I certainly think that we can. So one of the methods that we're using is read depth from whole genome Illumina data. And so basically, we take Illumina data and map it back to the human reference genome, and we look for regions in which we have a buildup of a higher copy of a particular gene. And so you could imagine doing the same thing for mouse, and you'd have to compare. You could look across different strains of mouse or species of mice, and basically you can do a comparison to look for genetic differences that way. So that's certainly one thing you can do. And there are back library resources, and so for mouse, and so you could also do the systematic sequencing of the regions that we've done for the mouse, for sure. But I think to begin with, you need to hone in on the region specifically, and that would be by doing these read depth estimates across the whole genome and looking specifically for regions that were different in copy number as a starting point.
Right, but when you already have the data as it is now with the assembly, I guess I'm asking whether or not you see regions of the mouse genome.
I see. You're asking me if there are regions in the mouse genome that look like this? So I haven't looked at that at all, and I'm not sure. I'm sure people have, but it's not something that we focused on in our lab. We do look at duplications of other mammals, but it's not something that we focused on. We certainly know of, the duplication landscape in mouse is quite different compared to human and other great apes. So in human and great apes, we've experienced this kind of this increase in intrachromosomal duplication that contain large genes that have these core duplicons that are on the edges of them. And this is really unique to the great apes. And so when we look within mouse genomes and other mammalian genomes, we see instead that there aren't these large-scale intrachromosomal events that kind of pick up whole regions and move them all the way to a completely different spot, but typically we see tandem duplications. And so this whole core duplicon phenomenon seems to be specific to the great apes. And so this has been looked at in mice. But if you were looking for other duplicated genes, you could use similar methods in the mouse. Yes. Yes?
Marla.
So are there examples of migration diseases, let's say in I guess chimpanzees, that is there less frequency of migration-like diseases in chimpanzees than there are in humans? So are there hydrocephalus chimpanzees?
Yeah, that's a good question. So I can certainly tell you that I don't know the answer to that question.
The question.
Oh yeah, sorry. So is there a reduced prevalence of neuronal migration disorders in chimpanzees compared to humans? Is that what essentially, and is that because humans have neuronal migration genes that, so I don't know the answer to that, but I would assume that it would be, that we wouldn't really know. There certainly is an ascertainment bias in humans versus chimps. And so certainly humans on a whole have larger brains, and so there's that. But they still have the same genes that we have in terms of neuronal migration, but they just have maybe less of them, the duplicated ones. But yeah, I don't think that that's well known.
And I guess the idea is that
You
there's
might.
a correlation between having duplicates leading to being responsible for larger brains.
Right.
And also being associated with disease states.
Right.
Are there, a wider variety of disease states?
Right. Yeah, and I certainly think that, like humans have a larger swath of neurological disorders compared to, but I think it's an ascertainment problem. Like we really aren't looking for autism in chimpanzees. We're not looking for these things, and so, yeah, yes,
With regard to the GOLGA elements,
yes.
if they're sort of like the locks P of the human genome, is there a corresponding creed? Do you know if there are proteins that promote the recombination, or
Right.
sites more or less mobile for recombination?
So to repeat your question, so are the GOLGAs, is it like a Cree? And is it a lock? Yeah, is it like a LOX-P system? And so the mechanism underlying the GOLGAs it's still pretty unclear to us what exactly is happening. One thing that we know for certain is that the GOLGAs, these high copy duplicons always have a highly expressed gene underlying it. So there's something happening there. Maybe it has something to do with open chromatin during meiosis, that maybe there's a conflict between transcription and also replication. It could be the underlying sequence component of the GOLGA. There does seem to be kind of a strange repeat structure within the gene itself that could be causing hairpins, for instance, and things like that. So it's unclear the precise mechanism by which these duplications are occurring via the duplicons, but it's something that we're trying to get at. Yes.
Any other questions? If you have questions, please raise your hand so we can get you a microphone. I have one last question. You had a case, I forget which element it was, maybe it was the beta element,
Yes.
where it looked like there was the most prevalent copy number was 7,
Yeah.
and there was a little bit of 6.
Yep.
Do you see evidence of, and I was trying to get my mind around that, do you have any evidence of balancing selection or cases where
Right.
the heterozygote is preferred or more common? I don't know how that could happen.
I know. So every time I see an odd number on the copy number prediction, I'm always like, why is this? And so in that case, I don't think it's a matter of actually the heterozygous state being more prevalent. I think it's a matter of there being a core duplicon within that alpha that was messing up our timing estimates. And so we were using the whole thing. And also it's a matter of the alpha duplication, what we called the alpha duplication, at other regions of the genome, it was partially duplicated. And so what we're getting is we're picking up partial duplications of alpha. And so it looks like it's like 6.5 or 7. And in fact, it's probably more like 8 is the overall copy number, but it's like a partial duplication
I see.
elsewhere. And that's what
Two
we're-
halves make up that extra odd number.
Exactly.
Great.
So that's what we're seeing. Yep.
Well, thank you, Megan.
Great.
That was
Thank
great.
you so much.